It was in 2020 that two spectacular women won the Nobel Prize for discovering the best gene editing technique, CRISPR-Cas9. This technology has revolutionized biotechnology by proving to be more efficient, practical, and inexpensive, as well as showing promise for the treatment of genetic disease. Hello, welcome to Genetic Curve by the Future of the Brain, a science documentary focused on advances in gene editing. In episode 1, we will discuss more about the concept of gene editing, CRISPR-Cas9, and how it can effectively help to treat Huntington disease. Let's go.
At first, it's necessary to understand that DNA is the molecule that carries the genetic instructions for the development and functioning of all living organisms. It has the shape of a double helix, composed of two interconnected strands, similar to a twisted ladder, with the rungs formed by pairs of nitrogenous bases, adenine A with thymine T and guanine G with cytosine C. DNA is essential for heredity, reproduction, and protein production.
In this context, gene editing emerged as a technique which DNA organism is modified, potentially altered, added, or removed gene sequences.
One of the many applications of the discovery is to treat or prevent genetic disease, such as the neurodegenerative disease called Huntington.
Huntington disease is a neurodegenerative, genetic, and hereditary disease that affects the central nervous system. It manifests as a combination of motor, psychiatric, and cognitive symptoms.
As a potential means to prevent the disease, CRISPR-Cas9 has been extensively researched and laboratory tests are already underway.
It consists of a guide RNA that directs a protein called Cas9 to the correct DNA sequence that needs to be edited. Cas9 function is to cut this mutant sequence that causes the disease, and after that, this RNA replaces the cut DNA strand, thus removing the problem.
But how does CRISPR-Cas9 actually help with Huntington disease?
Huntington disease is caused by an inherited genetic mutation in the ASTT gene located on chromosome 4, which causes an excessive and abnormal repetition of the CAG DNA sequence. This repetition leads to the production of a mutant and toxic Huntingtin protein.
In 2016, Shin and colleagues created an less selective CRISPR-Cas9 that approach capable of disabling only the mutant version of the HTT gene linked to Huntington diseases.
Young and colleagues in 2017 used CRISPR-Cas9 to non-specifically remove the HTT gene, reducing the neurotoxicity and the inflammation and improve the motor coordination in the mice.
In 2019, Ekman and colleagues inactivated the mutant ACT gene, reducing toxic agreement by 50%, prolonging life and reducing motor difficulties in test organisms.
Therefore, despite the existence of complications and challenges, both technical and ethical, the gene editing method shows great potential for treating genetic diseases, thus revolutionizing medicine and science.
Who knows what the future holds, right?
In episode 2, we are going to dive deeper into the ethical and moral dilemmas that surround the gene editing and the technical challenges applying this technology in humans. See you in the next episode!